All right. Um, Now it's my pleasure to introduce to you Irene Ong. Irene, I'm going to ask you 5 questions that I ask of everybody. You don't have to answer them, or you can answer them any way you would like. Those 5 questions, I'm going to tell you what they are. Where were you born? Where'd you go to high school? Where'd you go to undergrad and what'd you study? Where'd you go for your advanced degrees and what'd you study? And when did you come here to UW-Madison? So, Irene, where were you born?
I was born in Malaysia,
Where in Malaysia?
in Kuala Lumpur, the capital city.
The one city I've heard of. What does Kuala Lumpur mean? Does it have a name? Does it
it's
translate
it's like
in
muddy
Malay?
river
Really?
because it's
Wow.
near a river. Yeah, that's quite muddy.
Is it a trumpet player or anything like that? Or no? Um, Thank you. Bad joke. Moving on. And where'd you go to high school?
I went to high school in upstate New York. That was
Oh,
where
where?
when I moved literally from Malaysia and started school in upstate New York in Governor, New York. So
Governor.
really, really north, very near the Canadian border.
Holy cow. So it's up above, is it Plattsburgh?
Yep, above there.
Okay. Wow. Great. And
Yep.
where'd you go for undergrad?
I went to Cornell University for my undergrad.
And you studied?
I studied computer science and biology.
Oh, well, computer science is pretty easy. Biology, that's hard.
Yeah.
And where'd you go for your advanced degrees?
I actually came here in 1999 to do a PhD in computer science, and then stayed here because I really love Madison. So it's a
Great.
great place, and people here are wonderful. So,
And tell us what your location and job are now.
oh, right. I'm an assistant professor in OBGYN and biostatistics and medical informatics. Um, and I like to analyze biological data, systems, and as well as medical data, or, EHR from electronic health records, to try to find ways to improve people's health. So
Wow.
we'll see how it goes.
Not
Yeah,
bad for computer science. That's amazing.
trying to. It's not easy because there are lots of regulations, and getting access to the data requires IRB approval and all those other things. So working a lot with physicians and
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 where is your lab or office located?
my lab is an office is at the Wimmer Wedge, the new
Okay.
building. It's temporary, but it's in the new addition that's supposed to be part of the Center for Human Genomics and Precision Medicine.
Good. And do you have appointments in like the new data science and institute or any other things like that? It's impressive to me that you have a PhD in computer science and you have an appointment in OBGYN.
Thank you. Yeah, yeah, my life has taken me in, various ways, I guess, various routes. And I stayed on after I did my PhD, and did a postdoc with the Great Lakes Bioenergy Research Center, and then moved over to the Cancer Center, where I worked as a scientist for a while, sort of analyzing biological data. And a collaborator who I had worked with in OBGYN said, "Hey, I have the job for you," because they were looking for someone who knew genomics. And they said, "Well, she's even better because she knows genomics and machine learning." So they invited me to apply for the position. And
That's a great story
so
unto
that's where I
itself.
am.
So
So
congratulations.
thanks.
So today you're going to talk about something that's pretty cool to me, and that's the whole idea of how to antibodies bind to viruses. And it's been 35 years since I spent any time on this. I bet things have changed a little, so I'm looking forward to hearing what you have to say. Thank you, Irene,
Okay.
for coming and talking with us.
Thank you.
If everybody could mute at least, and then if you want to turn off your video, that'd be great. And then we can pop in and ask questions at the end.
Great. Okay, so thank you all for being here for this virtual presentation. I'll tell you a little bit about our work in characterizing the landscape of anti-SARS-CoV-2 antibodies in humans and cynomolgus macaque models. A year ago, I was working with Chris Surgi, Jim Gurn, and Palmerberg on a project involving analysis and integration of various data from a cohort. And one component was to profile and analyze antibody binding to human rhinovirus from children. And I had the array all completed, but then the pandemic occurred and labs were shut down. And I wasn't able to access the samples. So I thought, well, instead of sitting here and not doing anything, and being at the time was kind of scary and not knowing, how long it would last. I thought, why not profile the antibody binding to coronaviruses instead? So my lab worked closely with David O'Connor and Miriam Shaleff's labs to translate the skills that we had been using for research to identify antiviral antibodies produced in SARS-CoV-2 infection instead. And SARS-CoV-2 is the virus that causes COVID-19. So first, I would like to acknowledge everyone on the team. I'd like to highlight a rising star, Anna Heffron, who, along with others who did the work, David Baker, Sean McElwin, Maya Amjadi, and Sanya Kular. I'd also like to thank my collaborators, David O'Connor and the rest of the O'Connor lab members for their virology expertise and insights, Miriam Shaleff and the Shaleff's Labs biobank crew who provided the human samples, Yoshi Kaoka, Tammy Armbrust, and Peter Hoffman from Yoshi's Labs for performing neutralization assays on the samples, and Ajay Sethi, who helped with statistical considerations. And Palmerberg and Rob Kirchdorfer were helpful with the structural work. And then Ian Crozier, Courtney Finch, and Jans Kuhn are from the IRF Frederick, which is part of NIH. They provided the macaque samples that we profiled. And then we also like to thank the staff and scientists at Nibble Therapeutics who generated the peptide arrays and using the samples that we provided to them. So as an outline for this talk, I'll start by describing a little background on sort of immune cells, and then what we already know about anti-SARS-CoV-2 antibodies. And then I'll describe the methods we used in the study. So first, some background. Okay, so our immune system is comprised of innate and adaptive immunity, whereas the innate immune system is sort of pre-programmed to react to common broad categories of pathogen. The adaptive immune system is highly specific to each particular pathogen that the individual has encountered. So adaptive immunity creates immunological memory after an initial response to a specific pathogen, as shown here, either a viral or bacterial cell. And then that leads to an enhanced response to future encounters of that particular pathogen. The cells that carry out the adaptive immunity immune response portion are B cells and T cells, but we'll only focus on the B cells here. When B cells bind to antigens on the surface of pathogens, as shown here, they become activated and begin secreting antibodies. And what's shown here is a version of the antibodies, which are also called immunoglobulins. And they're the ones that are shown here are IgM, immunoglobulin M. And usually IgMs are produced first after an infection, several weeks after an infection, and then later, as you progress to the third week, you then see higher production of IgG, so it's immunoglobulin G, and those are usually what are more effective, but having some sort of protection is helpful. So antibodies travel through your bloodstream once they're produced. So sorry, the picture doesn't look as good displayed here, but these are supposed to be antibodies that are produced by the B cells, and they travel through the bloodstream and bind to the foreign antigens that those specific antibodies recognize, and that can help it to inactivate the pathogen, whether it's a virus or bacteria, and prevent the antigen from binding to the host. So, and antigens, you might ask, what are they? They're any substance that can elicit an adaptive immune response. So it has to be very specific, that structurally, sort of like a lock and key, that's recognized by either a B cell or T cell. But in this case, we'll just talk about the B cell. This process of adaptive immunity is really the basis of vaccination, and adaptive immunity can provide long-lasting protection, sometimes for the person's lifetime. Can anyone think of an example of a long-lasting immunity?
Measles.
Absolutely. Yep. Yep, that's right. But then there are some cases where people, so people who recover from measles are then protected against measles for their life, right? And in other cases, they don't protect over the lifetime, such as chickenpox. But coronaviruses, we're still studying, but from knowledge of other common cold coronaviruses that aren't as, I guess, doesn't elicit such bad response from people, they don't tend to last a lifetime, but they do stay around for a little bit, which is why these vaccinations can help. So, okay. What is currently known, our understanding of serology for SARS-CoV-2 infections are mostly limited and mostly centered on the spike or S and nucleocapsid or N proteins. We know that antibodies directed against the spike protein can be neutralizing, since the spike protein is what binds to the receptor on human cells, that leading to fusion with the cell membrane and introduction of the viral genetic material into the host cell. We know that anti-nucleocapsid antibodies are robustly produced, but they're not neutralizing. The nucleocapsid protein being the one that sort of binds and packages the viral genetic material. And those two proteins, the spike and nucleocapsid, are basically the only two that are employed in every vaccine and serology-based diagnosis to date that we know of. Though we know that there are two other proteins in the virion structure, the membrane, or M, and the envelope, or E. And additionally, we know that there are a number of other accessory and non-structural proteins that are synthesized by the SARS-CoV-2 virus, which the genome is shown below here. And so we were interested in profiling what those might look like, what the antibody binding to those proteins would look like. So numerous computational studies have tried to predict epitopes in SARS-CoV-2 based on what was known about antibody binding in other coronaviruses. So back in April 2020, Dr. Alba Grifoni from Dr. Alessandro Setti's lab predicted B-cell epitopes. So these are regions on the virus that are recognized by the B cells in SARS-CoV-2. By considering the alignment of B-cell epitopes in other coronaviruses with the SARS-CoV-2 sequence. And shown here are their predictions for B-cell immunodominant regions for SARS-CoV-2 in the spike, membrane, and nucleocapsid proteins based on SARS-CoV-1 specific epitope mapping. And basically the regions that are higher are indicative of positions where there are more antibody binding, and the numbers from left to right are the amino acid position for each of the proteins that are shown here. So other studies have also indicated antibodies to coronavirus proteins besides spike and nucleocapsid may be important. Several studies in SARS-CoV version 1 and in transmissible gastroenteritis virus, or TGEV, demonstrated neutralizing antibodies produced against the membrane protein. And then experimental vaccines using membrane envelope and ORF8, which is another protein in SARS-CoV-2, can elicit neutralizing antibody responses. And additionally, antibody-dependent enhancement, in which the presence of antibodies actually enhance the disease rather than protecting, has been shown in a number of coronaviruses, sometimes implicating antibodies that are produced against the spike protein. So there's a lot we don't know, and that's why we wanted to profile the antibody binding across the whole landscape of the SARS-CoV-2 proteome. So now onto our methods. First, our study population. Our human samples were collected from 40 COVID-19 convalescent patients and 20 naive controls. These individuals had varying ages, severities of disease course, comorbidities, and so on. They donated samples 5 weeks after symptom resolution, which means time since infection is variable among the group, since that time will be much longer for a person who is hospitalized on a ventilator than for a person who had a mild illness and was never hospitalized. But this variability does make it a little easier to translate our findings to the real world. And in data I won't go over today, we were able to make comparisons among the clinical severity subgroups. The human subjects had a variety of presentations from very mild symptoms out to an extended amount of time on a ventilator. About 40% of our subjects were hospitalized for their symptoms, while the other 60% of our human subjects were symptomatic but never needed to go to the hospital. From the animal side, we profile a population of 3 macaques that were infected with SARS-CoV-2. All were young and healthy, and they all underwent intrabronchial instillation of SARS-CoV-2. And in this study, they used serum samples that were collected 11 days prior to infection, and then 12, 19, and 30 days post-infection. So from multiple macaque studies, we know that macaques develop similar lung abnormalities to those seen in humans, such as the ground glass opacities in a crazy paving pattern seen above in these CT images, with a human COVID-19 chest CT image shown on the left, and the macaque COVID-19 chest CT image shown on the right, with comparable lung disease shown in the red and blue boxes respectively. Macaques have not been observed to develop severe disease, only mild and moderate. And additionally, macaques are known to develop binding and neutralizing anti-SARS-CoV-2 antibody titers. So all 3 infected macaques developed antibody titers as detectable by binding ELISA, as shown here, at least by day 12. And animals V2 and V3 developed neutralizing antibody titers, whereas animal V1 did not. And you can also see the 3 control animals that were used in the study that were mock infected, macaques M1, M2, and M3, but that they did not produce any detectable antibodies against SARS-CoV-2, so we did not profile their samples in this study. So for this project, we used this ultra-dense peptide microarrays from Nimble Therapeutics, which is a local startup company. These arrays involve synthesizing peptides from protein sequences of interest, and in this case, we used our coronaviruses, and then synthesized each peptide 1 amino acid at a time on a surface of a slide. The array is 1 by 3 inches large and holds up to 6 million unique peptides for 1 sample, or 390,000 each for 12 samples. We used the latter arrangement, and we used peptides that were 16 amino acids long, because this is what we saw had the best results using this length. And each peptide is tiled in tens or thousands of copies. And so it's essentially like performing 390,000 ELISAs per sample. You might ask, what is an ELISA? But I'll go over it very briefly now. So epitope identification using this tool involves taking a protein here. So for example, considering the coronavirus spike protein and breaking it into 16 amino acid peptides that are offset by 1. And so this is showing the 16 amino acid length and then how it's shifted over by 1 offset. And so we take these peptides and we tile them on the array, and an individual serum is then run on the array, and the primary antibodies from the serum bind any peptides that they recognize. And then a secondary antibody is then applied to bind to those primary antibodies. And we measure the fluorescence intensity of that secondary antibody to determine where antibodies have bound peptides or epitopes or antibodies from the serum. And then we look for binding in multiple contiguous overlapping peptides, such as this one, because its neighbors also have strong binding, as shown by the sideways plot there. Because we think that it's more likely that it's a real binding as opposed to a single spike in fluorescence that could be due to spurious binding or an artifact. We look for this binding in consecutive peptides throughout the proteins that we're interested in. And in this study, we look at longer term immunity against IgG antibodies. So some caveats: using this assay, we can only look at linear epitopes. We don't know whether it can detect parts of conformational epitopes. And of course, it says nothing about the functionality of the antibodies that we observe, rather only measuring that they can bind. And so if you look at it from the whole protein perspective, this is what it would look like for an individual sample against a whole protein. Along the x-axis here, we have the amino acid position of the coronavirus spike protein starting at 0 on the left and going all the way out to the final amino acid on the right. And then the y-axis shows the log normalized fluorescence intensity for any individual serum sample. For this array design, we tiled hundreds of proteins, including many full viral proteomes on the array. We included the full proteomes of all coronaviruses known to infect humans. And so our SARS-CoV-2 at the top, and then SARS-CoV-1, the Middle Eastern respiratory SARS virus, and then the 4 human coronaviruses, HKU1, OC43, 229E, and NL63. Those are your common cold coronaviruses, as well as the full proteome of the bat and pangolin viruses, because those are actually much more closely, they're large sequence similarity with our SARS-CoV-2 proteome. And then we also included the spike proteins from a number of more distantly related animal coronaviruses. And then additionally, we included 248 SARS-CoV-2 isolates that were collected in Wisconsin prior to April 24th, 2020, which would have been the time that some of our human samples were infected, but I will not be talking about those today. We also included a number of proteins as controls for our human samples, since we would expect that all human adults have been exposed to and have a response against at least one of these proteins. And all of these were fit onto a small section of the chip for each individual sample. So to visualize the representative strains here and what their relationship to SARS-CoV-2 would be, so the percentages are indicative of percent similarity based on their sequences. So we included the SARS-CoV-2 is right there, and we included the beta coronaviruses that infect humans, and those are shown in blue. And then the alpha coronaviruses that infect humans are shown in salmon, and the animal coronaviruses not yet known to infect humans are shown in yellow, and these include coronaviruses known to infect birds, rodents, pigs, bats, and pangolins. Okay, are there any questions so far? feel free to jump in if you have any questions. But okay, if not, I will go on to what we found. So I'm going to break our results into 4 parts. The epitope identification, basically where the B cells are identifying regions on the viruses that they're binding to. The macaque model comparison to humans, cross-reactivity across different coronavirus response, and then the new variant strains and how they overlap with what we have seen so far. So I'll start with the epitope identification. So first, as validation, we wanted to ensure that our methods were capable of picking up known or expected responses. So we wanted to look at our control proteins, and here is the poliovirus polyprotein. And you can see that along the x-axis, the amino acid position in the poliovirus polyprotein. And then along the y-axis are our individual samples. There is a horizontal line across this plot or heat map, I guess, if you want to think about it that way, that's representing the binding for each unique individual, with the 40 COVID-19 convalescent patients up at the top 2/3, so each of them horizontally, with this black bar shown here. And then the 20 uninfected controls from prior to 2019 in the bottom third of this indicated by this gray bar. And so in this heat map, we're showing the log2 normalized data, with blue representing the least amount of fluorescence intensity or binding, and red representing the most fluorescence intensity or binding. And the black and gray line plots at the top represent the average fluorescent intensity for each group, the COVID exposed or I guess COVID infected patients versus the controls. But they're so similar that they actually overlap each other, and they look like the same line plot. To call positive antibody binding, we used a cutoff of fluorescence intensity that is 3 standard deviations above the array data. And what we see is that in every one of the samples, both patients and controls showed antibody binding at a known linear epitope in polio, starting at around amino acid 620, which we have also observed as a positive control in other human samples, validating that the assay is detecting binding. And okay, so to call the SARS-CoV-2 epitopes in our human samples, we use slightly more discriminatory standards than the 3 standard deviations above the array mean, in an effort to remove some of the probable cross-reactive binding from previous coronavirus infections in this human population. And so we required 3 conditions: that the epitopes be greater than 3 standard deviations above the log2 quantile normalized array data. They're also greater than 2-fold reactivity compared to controls, and that they were at least weakly significantly different from controls, using a two-sided t-test with unequal variance and Bonferroni-Hochberg adjusted p-values of less than 0.1. These methods allowed us to identify 78 B-cell epitopes throughout the SARS-CoV-2 proteome, and these epitopes indicated by orange shading in the line plots. And I'm going to go into each a little bit more in detail. The x-axis shows the amino acid position in the SARS-CoV-2 spike protein. We're now looking at the heat map of the spike protein. I know that all the heat maps look the same, but this one is looking at the spike protein. And the individual human samples are stacked vertically. Again, the black bar on the left indicate the COVID patient samples, and the gray bar along the left are the controls. Blue indicates little to no binding, and red indicates the most binding. And you can see here, and again, the line plots at the top represent averages for the patients and controls. And then the orange shading represents areas in which we identified an epitope, meaning that there were much more antibody binding for the COVID infected patients at that position of the virus than for those who were controls. And so we can see a lot of areas that are lit up as epitopes. And in particular, we can see a lot of reactivity adjacent to the receptor binding domain, which basically ends at the amino acid 541 position. And particularly in the fusion peptide of the spike, which is around 780 to 810 here. And then also in the haptat repeat 2 region, which starts around 1150. And both of those two, the fusion peptide and the heptat repeat 2, which start are involved in membrane fusion for the virus. Interestingly, we saw very little reactivity actually in the receptor binding domain, which is what actually binds to the cell receptor on the human host cell, which stretches roughly from amino acid 320 to 541. As an exciting validation of the robustness of our methods, we were able to detect 4 instances of binding to regions associated with neutralizing antibody activity indicated by stars that were previously documented by others in the literature. And then just to be able to visualize what it means in a 3-dimensional structure, because a lot of the data I've been showing is more in a 2-dimensional representation. This is looking at the structure of the SARS-CoV-2 spike protein with blue to red scale for least to most binding. And so we saw some of the highest binding in spike in the fusion peptide, and then very little binding in the receptor binding domain. And yeah, just so that, all of this is actually happening in 3-dimensional as a reminder. So moving on to the other SARS-CoV-2 proteins, again, with amino acid positions along the x-axis, and each individual patients in the horizontal line, patients in the black, and then the controls are gray. We see quite a number of epitopes indicated by orange shading here as well. And there are epitopes in the ORF3A protein, in the membrane protein, ORF6, ORF8, and the nucleocapsid. And we did not detect any epitopes in the envelope protein, which is perhaps not terribly surprising, because even though it's a part of the virion structure, it's a very small protein, and also epitope prediction methods did not predict epitopes in the envelope. Particularly interesting was the membrane protein, in part because one of the epitopes in this protein had the highest combined reactivity, sensitivity, and specificity of any that we recorded. Additionally, while you can see, as in the nucleocapsid here, and as was the case in the spike, most of the proteins we assayed showed quite a bit of reactivity in the controls, but for this particular membrane epitope, there was minimal activity in the control. As you can see, there's much more red at the top 2/3, and then it's mostly blue in the bottom 1/3 where the controls are. So yeah, zooming in, considering this membrane epitope at the N-terminus of the membrane protein, we see high reactivity in the patients and very little in the controls. So M proteins are the most abundant proteins in the coronavirus virions. The N-terminus of the M in other coronaviruses is known to be a small glycosylated ectodomain that protrudes outside the virion and interacts with the spike, nucleocapsid, and envelope proteins, while the rest of M resides within the viral particle. Though membrane protein has not been investigated much in other coronaviruses, in one study, the injection of the full-length SARS-CoV-1 membrane protein was shown to induce neutralizing antibodies in rabbits. And then they were found to be more strongly neutralizing than anti-S or anti-spike antibodies, and also to have synergistic effect with anti-spike antibodies. Looking at binding protein for antibodies against M in SARS-CoV, again, from the predictions paper, we see that the patterns we observe matches that of those in the SARS-CoV-1 And however, membrane protein, again, still remains largely understudied and underutilized as an antigen. So Maya Amjadi in Miriam Shalef's lab also performed a validation experiment using ELISAs with some of our highest performing epitopes in spike, membrane, and nucleocapsid. These results confirmed our peptide array findings. And what was really striking was that while there was some reduced specificity in the two spike protein epitopes we tried, the 1253 spike position and the 814 amino acid position in spike, the membrane and the nucleocapsid epitopes, membrane at position 8 and then the nucleocapsid at position 390 remained highly specific. As you can see, the controls are really low, and there's a separation between the COVID-19 patients and the controls. So, okay, now that we've established the landscape of the antibodies in human SARS-CoV-2, we wanted to determine how well this carries over for macaque models and see how well they compare. So again, I guess in the interest of time, I could probably go through this quickly. The x-axis here is this again, the same amino acid positions for SARS-CoV-2 spike protein. And we're looking at longitudinal samples for the three macaques, V1, V2, and V3. And each horizontal bar represents a single time point from each animal, starting with pre-infection at day 11, I guess 11 days before the infection, and progressing over time to 30 days post-infection. In these heat maps, again, blue is the least amount of binding and red is the most. Okay. We clearly see no or minimal reactivity at the pre-infection time point, with reactivity appearing in all three animals by day 12, as shown here, and continuing to increase through day 30. We see some good shared signals, especially around amino acid 800, corresponding to the fusion peptide in spike, and then another shared signal out at amino acid 1150 in heptat repeat region 2, again, both involved in membrane fusion. Now looking at a comparison with the human patient's results, I'm going to start with just the day 30 time points, as that is the latest time points to compare with our much later human sample time points. Even at these early 30 days post-infection time points in macaques, we see binding to epitopes we identified in humans, which included 3 out of the 4 epitopes that have been shown to be associated with neutralizing antibodies. Filling in the rest of the longitudinal data, we can see in the macaques how binding to these epitopes sort of increases over time. And if you'll remember that the macaque animal V1 here, it did not produce, or she did not produce any detectable neutralizing antibodies, which also happens in humans. That phenomenon of producing some antibodies, which apparently no neutralizing capacities in them. But here we can see she actually produced antibodies to multiple epitopes that multiple other groups have found to be neutralizing. So this is quite intriguing. Maybe she produced these antibodies at such low levels that they couldn't neutralize, or maybe it's possible to produce antibodies of different sorts against these epitopes, and V1 just didn't produce them. And it might be a titer issue, meaning how much of that is produced. And that the titers for this animal basically weren't produced high enough to neutralize, which would also help explain why it's not uncommon to see this effect in humans as well. Okay, so now looking at the 3 macaques longitudinal data, but looking here only at the proteins for which we had detected epitopes in humans, ORF3A, membrane, ORF6, ORF8, and nucleocapsid. The x-axis is the amino acid position in each protein and each time point from pre-infection through day 30 for our 3 animals. We also see some responses arise after infection that continue to increase in magnitude for these other proteins as well, besides the spike protein. And then again, particularly our binding in the membrane N-terminus region of the membrane protein. So comparing to humans, we see some alignment with epitopes for some animals in nucleocapsid and alignment for all animals of the membrane epitope, which also lines up longitudinally, confirming that antibody binding patterns in these macaque models effectively mirrors what we see in humans. Okay, I guess I will skip through that. And since we're running out of time, I will quickly go over the cross-reactivity and how these antibodies bind in other coronaviruses. So what I'm showing here is just the human data now. And we're looking at binding for the 7 human infecting coronaviruses, including 4 beta coronaviruses in blue and 2 alpha coronaviruses in red, and a bat and pangolin coronavirus in yellow. And with all of these other viral protein sequences aligned to the sequence of the SARS-CoV-2 sequence. That alignment is why you see some white spaces where there are gaps due to the alignment. So here we're looking at the averages for the human patients with the black bars, and compared to the average of the uninfected humans in the gray. We use the same 3 discriminating standards as I mentioned before as a way to call reactive epitopes. We saw several cross-reactive epitopes, with the most cross-reactive ones in the spike proteins of all of these viruses here at the amino acid position 800, corresponding again to roughly the area of the fusion peptide, and one epitope which was cross-reactive for all beta coronaviruses considered at amino acid 1150. Heptat repeat 2. So both of these, again, being verified as neutralizing antibodies in SARS-CoV-2 infection, and both areas of the protein being involved in membrane fusion. There was additionally cross-reactivity in other proteins that was mostly limited to cross-reactivity in SARS-CoV-2 and or MERS-CoV in terms of human coronaviruses, as you can see sort of in the middle at the top there, and but not any other human coronavirus, which is itself really interesting, that we do not see cross-reactivity throughout the proteome for the deadly human coronaviruses, the SARS, MERS, but these 2 epitopes in spike were the most broadly cross-reactive. They are the only ones that were universally cross-reactive across at least all beta coronaviruses. So having established that, I think I will skip these. This one is, which is basically looking at responses to spike across the different other animal species, as seen in the macaque data. I will go on to the variants of concern now, and as you've probably heard, the variants of concern in SARS-CoV-2, as the virus has been passing widely around the globe, it keeps mutating, and a number of these mutations seem to give certain variants a selective advantage. The CDC has identified 3, and I guess recently it's now 5 known variants of concern that are shown here. The 3, so B.11.1.7 was first identified in UK as believed to be transmitted more effectively, while B1351 and P1, first discovered in South Africa and Brazil/Japan, respectively, are believed to be more efficiently transmitted and resistant to some antibodies. And they are shown next to all their characteristic mutations to the right here, shown as protein colon mutation. And we wanted to see how our antibody findings matched up to these variants that were detected in these strains. So those mutations that appear in red boxes are in areas where we detected antibody binding, and those in blue ovals are within 3 amino acids of where we detected antibody binding. Our methods predicted about half the mutations in the B117 variant and the B1351 variant in spike and other proteins, but a lot less in the P1 variant. So what does this mean? That these individuals who gave these samples in May to June had antibodies that correspond with a lot of the mutations that have since arisen. It might mean that antibody escape is a selective pressure that's driving the emergence of these variants. So they mutate those regions where there happen to be lots of antibody binding. So a recent preprint from the Bloom lab have shown that antibody escape seems to drive evolution of another human coronavirus, the human coronavirus 229E, common cold coronavirus. And then the Hatziano and Binash labs also recently published a paper in eLife showing that that appears to be happening for the receptor binding domain of the SARS-CoV-2 spike protein. And knowing that in our data, we see binding in quite a number of epitopes that are not the spike receptor binding domain, these may be involved in mutation through antibody escape as well. So final overall conclusions. So SARS-CoV-2 infection results in robust, sensitive, and specific antibodies in structural proteins and non-structural proteins. Antibody binding in cynomolgus macaques mirrors that in humans, confirming their value as immunological models. Anti-SARS-CoV-2 antibodies consistently bind proteins in other coronaviruses, especially in S protein. And epitopes we identified corresponded with some mutations in emerging variants of concern. And our next steps include developing methods for identifying epitopes at the protein level, and looking at antibody signatures in children with common cold coronaviruses and adults with asymptomatic cases, and seeing if there are, cross-reactive antibodies that are protective against the SARS-CoV-2 virus, which might explain why children have milder, or some people have milder disease than others. And then also following up on the functional status of the antibodies that we discovered, to see if they have neutralizing capabilities. And also we know that my collaborator Miriam Shalev has worked with UW to use this membrane peptide to distinguish UW healthcare workers who have since been infected post-vaccination. Because if you use your regular testing after your vaccination, it seems like you've already had COVID, but using this method of just looking at the membrane peptide allowed us to distinguish between those who were vaccinated versus those who have since gotten the infection post-vaccination. Again, I want to thank everyone who helped with this project and to point you to the preprint, which we've put some of our early human findings in greater detail on bioRxiv. I'll also say it's been a privilege to work with my collaborators on this emerging pandemic as it's occurring, and gratifying to be able to apply methods to this outbreak while it's ongoing. And thank you for your time and attention.
Thank you very much, Irene. So if you'd like to ask a question, you can unmute. You can also show your camera if you'd like, and ask Irene a question. I'll start, if I may. You were doing polyclonal antibodies on this, is that correct?
They're exactly because they're antibodies from humans. Yep.
And what, if anything, could monoclonal antibodies show or not show that the polyclonals that they're working on?
So if we were to isolate the membrane epitope, antibodies that bind to that epitope, and make monoclonal antibodies of those, and show that they're able to neutralize the SARS-CoV-2 virus, then that could potentially be used as treatment for some people who are unable to make their own antibodies because of, either chemotherapy or that they're immunocompromised. But I don't know if I answered your question or not, Tom.
No, I'm just trying to keep track of what kinds of antibodies were being used. I have a can of WD-40 here and I'm spraying it on myself because I'm pretty rusty. But that's what I'm trying to figure out, what you can get from this kind of cool epitope mapping. Other questions for folks?
But yeah, that's a great question, because it is just looking at, so antibodies have two ends in which, right, because it's a Y shape. And so this side can bind to something and that side can bind to something else. And you normally, when you think of the protein as a three-dimensional structure, you could potentially have this binding somewhere and this binding a separate position, which is what Tom is referring to as polyclonal, meaning that it's binding to two different locations. Because the arrays are the way they are and it's flat, and we only are probing for linear epitopes, essentially all of those antibodies are only binding in one position, either one end of the Y or the other end of the Y, to those peptides. And so in that sense, it's a monoclonal probing of the antibodies. So
other question?
Great question.
Thank you.
Yeah, I had a question about what portion of the spike proteins on a virus need to bind with antibodies in order to effectively neutralize it as an infectious agent.
That is a question that we don't have an answer to, right? And that was one of the questions that we are interested in finding out. So antibodies can work if you just have one that's a neutralizing antibody that is able to either mechanistically prevent the spike protein from opening and closing, because it has to be in a specific configuration in order for it to attach to cells. If the antibody is, even if just one is binding in a very specific position that allows or that forces the spike protein to then be fixed, then it can no longer bind. So you could just have your, one very specific antibody target that could neutralize the virus. But in some cases, if the person doesn't produce that one specific antibody, then having the other antibodies with different epitopes that just sort of covers the surface of the virus, that can also help, sort of prevent the virus from making more copies of itself too. So it's hard to say, because it really depends on the specificity of the antibodies that are produced by each person. So
yeah, that's an impressive amount of data you're able to get out of your material. It's really, really impresses me.
Thanks.
Other questions?
Yes, I have a question. Can you hear me now?
Yes, we can.
Okay. I have a question. Does your research have applications for future vaccine development?
Yes, absolutely. Great question. And that was what I initially thought we would, be helping with if there were the progress hadn't been so quick in finding vaccines that work. So normal, I guess, there are multiple ways of making vaccines, and in some cases, you could just use the whole spike protein, which is what they've done with the mRNA vaccines. But then in other, and also the Johnson & Johnson and AstraZeneca ones. But I think with the Johnson & Johnson and AstraZeneca ones, where they're not giving it the mRNA, they're actually giving it the spike protein itself. But I believe those were fixed to be in a specific configuration so that they could not, bind to the human cells. And so that's what was used as a vaccine in the Johnson & Johnson and AstraZeneca vaccines. In in other cases where you want to minimize the cost of the vaccine production, you could just use these peptides where you know that it will elicit antibody responses from people. And so usually it takes many years to identify which are these specific epitopes that are eliciting the important responses, and that isn't going to cause side effects like what you're seeing in or hearing in about some of these like the AstraZeneca and the Johnson & Johnson cases. Because if you use everything, then there might be a case that the binding is similar to some other protein in your body, which is what we're seeing in some cases. Usually the body is able to remove those, right, that look similar to a protein in your own body, and that prevents autoimmune diseases and that sort of thing, and would prevent these side effects with the binding, with the platelet binding, or I guess with the blood clots. But which is why usually when people develop vaccines, they are careful to figure out which epitopes are the ones that elicit the good responses and leave out the ones that are the bad responses. I think the approach this time was, we just need something that works. And so that's why the approach was, let's just use the spike protein. We know there's a lot of responses to this, and we know that they can elicit neutralizing antibodies. So let's use the whole thing. But if we wanted to make this and make vaccines that are cheaper,
less expensive,
right, we could produce ones with these peptides and of course undergoing all the safety things, and then make them available to others who don't have access to these types of vaccines.
Thank you so much.
Yep, you're welcome. And it's also helpful for diagnostics. So as we were showing, you can identify those who have been exposed by looking at by using the peptide from the membrane region, and seeing if the antibodies from patients who have been exposed to COVID can bind. And if they do, then you can say that they've been exposed to SARS-CoV-2. So.
Awesome. Are there any other questions? All right, well, it seems like we're kind of coming to an end. Thank you so much, everyone, for coming. Thank you so much, Irene, for the awesome talk. If anybody has any more ideas for Plato and what to do next semester, make sure to email Tom, and his email is still in the link, or is still in the chat. But I think that is everything for the spring semester, and that is everything for me. This will be my last Plato as well. So thanks for an awesome closing, Irene.
Thank you all. Appreciate it.
Thank you.
Thank you, Irene.
Thank
Wonderful
you.
research.
Thank you.
And thank you, Bea, for your work.
No problem. It's been a pleasure being with you all every Wednesday.
Thanks. We'll see you in September. Not you, Bea, but maybe you will.
Everyone else. Yes. Awesome. Thanks again, Irene. And I think we will close it up then.
Sounds
Thanks,
good.
Bea.
Take care. Yep. Bye.
Thank you.
Bye.